by session. So you can access each session and you will have slides, and sometimes also we shared codes for running some of the analysis that they are presented. And as another supporting material, you will have all the sessions in YouTube. So just have this in mind. And today, let me just stop sharing my screen. We will continue with this exciting series of talks, and we will have with us Dr. Ville Pimenoff from the University of Oulu. He's adjunct professor in evolutionary medicine in that university in Finland, and he leads an exposome cohort for monitoring exposures during pregnancy with a wearable device. Also, he is co-PI for the safe biomonitoring by biotic integrated wearables, and his main research lines include longitudinal population cohort epidemiology, population genetics of human microbiome, biotic and abiotic exposome in pregnancy, and wearable device monitoring. And today's talk, it's about comprehensive biotic exposome estimation from wearable devices metagenome. So whenever you want, Ville, the floor is yours.
Thank you, Augusto. So let me first put the presentation. Yes.
Perfect.
You can see it. Okay. So as Augusto said, I will give you a short summary of about biotic exposome estimation, and starting with the concept of a biotic exposome. So this is the concept of exposome, which most of you should be familiar with already. In this presentation, I'll focus on the biotic part of the exposome, which is the life course biotic exposures from prenatal period to elderly life. And as we know that the exposome is very comprehensive, here you can see the one of the well-known figures to describe the full exposome. I'll focus on the biotic one, which in its essence especially deals with the outdoor and indoor air exposures, what we are exposed to. It doesn't cover all of the concept of the biological exposures, but one, at least we think it's one of the most important part of the biotic exposure. So talk is focusing on the outdoor and indoor air exposures. And why this is, as you know already from the short intro in the presentation leaflet. So exposome is the accumulation of exposure during the life, and our idea is to develop a way that we could first of all measure this biotic exposure, and if it could explain some of the unknown fractions of the adverse pregnancy outcomes, as pregnancy defects has been studied for several decades, and we know a lot of important genetic and environmental stressors or cofactors, but there's still a large fraction of unknown factors, and we think that exposome, especially biotic exposome, could answer to some of these. And as already said, the key point is to measure these biotic exposures. And there, in the study I'll present here, is focused on using the ultrasonic personal air sampler, UPAS as an acronym, which is one type of a wearable device pioneered by the Stanford team, especially Snyder Lab, on trying to develop ways to measure and monitor longitudinally human exposures. And it's a device, which is a bit larger than a cigarette package, with a filter cartridge where you change the filter, which with the air pump of the UPass, you can accumulate biological matter and chemical matter. You control it by the mobile phone app, and as shown in the right-hand side, you can wear it on your waist or on the shoulder with different ways. We used this kind of shoulder bag for the pregnant women, as they said that was the most comfortable way to wear this. And this wearable device allows them to have a longitudinal biological and chemical profiling. And the biological profiling is based on the accumulating biological matter on the filter, which you then extract, and then you deep sequence the DNA RNA out of it. What I'm not focusing on the talk is the chemical profiling, but then added value is that the device also collects GPS coordinates, so you can always locate your results also in real life locations, so you're not dependent on the personal reporting. But this has a data-oriented restrictions, because most of this type of data is very sensitive data. However, those I'll cover also, like how to manage those. So based on all this, we started a proof of concept study here in Finland to monitor 100 pregnant and non-pregnant women for at least 4 months using the wearable device in order to have a proof of concept. I mean, 100 individuals, it's not enough to find the true causations, but to see that how feasible it is and what we can get out of it. And the actual monitoring part, I addressed that it would not be possible if you don't have a good study nurse team. Here's our team of 10 study nurses with whom we launched the monitoring across Finland in 11 cities, including the local healthy units, UPass training, and also having the support desk run by the nurses, and having the logistics of the sample collection. So once in a month, the sampling was done in a health unit, and the other time per month it was self-sampled at home with detailed instructions on how to pack the filter and then bring it to the next meeting in the health unit. And we in total recruited 102 individuals, from which 31 were pregnant women after the second month of the pregnancy. Here's the quick schematic diagram of the detailed sampling. So to add up from the personal aerosol exposure monitoring, we also collected the standard urine, genital swab, saliva serum, and also cardiac rate monitoring for those time flows, and the non-stop monitoring was basically done using the UPass device. And we just finished the 102 women follow-up for the whole cohort. But what I present is the pilot data of the 25 individuals, which we had 2 months follow-up, which we piloted on the whole flow of sampling, processing, and analysis. To go a little bit more in detail on the actual
saliva, buccal swab, then vaginal swab.
And then bit details on the filter processing. So the filter we get out of the UPass device we divide onto nucleic acid extraction, and then one part goes to chemical extraction. And so the DNA and RNA sequencing is then done on the deep sequencing method. But the critical part here is that it is very low biomass sample. So in that sense, the method and the sample flow needs to be very well standardized. And for that reason, we used through collaboration, we used automation on the library preparation, so that doesn't create the batches for individual work. And then also through collaboration, we had a possibility to use one of the latest sequencing methodology, which is Ultima sequencing, which is produces a little bit larger, high quality, low cost sequencing reads than standard Illumina sequencing. And at least based on the piloting data, the data quality looks very good. Apart from creating the data out of the wearable device, the next step is also how to handle the deep sequence data, because those are very large metagenome sequence files. In total, they're sensitive data. So in Finland, we have the possibility of using the Finnish IT Center of Science, CSC, computational resources, which are free for use for any Finnish researcher. And with them, we have developed a system to have a submission engine, so where you, as a cohort creator, can store the raw data, and then after you get it cleaned, published, and so forth, it is also possible to share with. But the main point here is that you have a secure infrastructure where you can handle these large set of data without problems on manipulation, storage, and later on sharing. What you should get out of the device in different aspects, and in this cohort, what we focused on the right-hand side is an example phylogenetic plotting. It's from the previous data, not from our cohort, done by our collaborators, but it shows individual variation of the phylogeny of taxa found in the filter. So this is one part. Then we have the GPS coordinates. These are two examples of the cohort where you have the exact locations, and another analysis is to then have the locations done with the cluster analysis, so that we have a bit of an idea of how variable are the locations within the cohort, which can have an effect, and it can be as a cofactor in the actual association of exposures. And another part which is interesting, which we discovered during the cohort, is that because we started during the pandemic, that because we also monitored the cardiac rate, so we could have an indication if someone has a infection, for instance, with a COVID, based on the cardiac rate baseline. And this has been already proved by the other data, but we can also see that there's some traces of COVID possibility to detect using these wearable devices. So it has interesting angles to look at for the exposure. And as I mentioned, there's also the chemical side of the wearable device, but I'm not going there. And this time, particularly because we haven't created the data yet for that part, but it is an added value then as based on previous data, we can see that the biological and the chemical has a very interesting correlations regarding the exposures and the effect on our health. But let's get back on the biotic profiling and why is it important. So if we think about human pathogens and how we how we are exposed to them and or how they infect us, because those are a little bit different concepts. The microbes which we exposed or infected, they basically involve with the host, so with the humans, which means that during time, the pathogens develop a lot of variation. And this variation is often shows different levels of pathogenicity. So some of the variants of the pathogen are more harmful than the others. And in that sense, the phylogeny of any pathogen species is important if we want to make estimates on the effect on our health. And as an example, here is a simple phylogeny of one viral family infecting humans only, and we know about 200 different strains of the viral family, but only about 15 strains actually cause any disease, and the others are commensal. So in that sense, if you only look at the viral species, for instance, that doesn't give you that accurate information, but the strain level information is important, or at least the phylogeny. The other side of the coin is that the detection of pathogen strain is not trivial. So there's several caveats that you need to find a solution in order to actually make a distinction on what you're looking at or what species or strains you can actually confidently detect. And one of the main reasons are simplified here is that when we look on a large variation of exposures, so biotic exposures, which we look from the metagenome, and this to have the resolution to know the strains is often not possible for most of the taxa, for the sheer fact that we always have a limitation of the data within the metagenome. So if we have in the metagenome viruses, bacteria, fungi, and a trace of human DNA, we simply do not have enough nucleic acid data to build enough strain level genomes so that we could have a high average nucleotide identity of all the taxa. But then you always have to choose on the level or layer that what you have in species, what you can detect as a subspecies or strains without talking about genomes when you work with the metagenomes. And this is a field of computational genomics, which is currently developing very fast, but we definitely do not have solutions for all, but it's in constant progress. And one of our ways to tackle this using the wearable device data is the multi-omics concept, where we do assembly using different tools, also direct taxonomic classification. Then we estimate on what are the reliable abundant taxa, so which we should use. And from metagenomes, so we build the metagenome assembled genomes, and with the direct species level classification, we can make an estimation that what is the true composition of different species and subspecies what we have in the metagenome. So this is as a simplified structure on how to estimate the biotic exposures from the devices. And here again, a simple example of a same sample, either DNA or RNA from the UPAS device, and where we did a bit of a benchmarking to see how different pipelines detect the different pan-domain taxa. So either using Megablast, which is developed by collaborators, or K-MER-based pipeline, which we have executed. So we get very similar level of classification. And as looking on the bacteria, fungi, or virus, they vary a bit, but that is very much expected when you have a different type of parameters. And this preliminary data basically simplifies the true fact. What we know already, work on the metagenomics, is that we don't have same criteria or method that would apply on all type of metagenome data or microbes. Also, we know that the reference data, so the known microbial taxa and the unknown knowns are constantly accumulating. So what you could do a year ago or a few years back is totally different than what you can do now with the known accumulating reference data. Then the true challenge is that how you can be reproducible with your analysis and work. And there, I think those ones who have an answer on this, it's that will be a very interesting to hear if you look on the environmental exposure, which is a lot more diverse than what you look on standardly looking microbiome in the human gut or the mouth. And for that, for instance, this nice known study on looking the microbiome signals using different methods and giving all different results. So these are the true challenges we face on understanding on environmental exposures. Another example, this is the paper done by or reanalysis done by Giavi on the microbial analysis on the TGGA data, which got critique on, and as the diagram shows, that if you use a metagenomic method, which not only detects but predicts the selection of the balanced phylogene based taxa found in the metagenome, which is the red bar plot on the left, and then the reanalysis showing that these taxa, which were predicted to find in the sample, were actually mostly not found, and their abundance was very different. As you look on the read numbers are very low, what was done and found in the reanalysis. And why I'm presenting this is because using the pipeline we have addressed on the UPAS metagenome data. So here we're just dividing the data they used in the original paper. The blue ones are the ones and the gray ones. The blue ones are high abundant, good candidates to be analyzed in the data. The gray ones are low abundance but good candidates, whereas the red and most of the yellow ones are likely false positive or background noise, depending on the classification method you use. We found this large fraction which should be excluded there because of not being able to verify all of it. And this quite nicely simplifies the or demonstrates the challenge how to analyze data which you get from very low biomass original samples, like in the UPass devices. So now I'll go into the pilot data set where we had 25 individuals. We had the filters for 2 months followed, so we have 4 time points. And these filters we processed, and we did the deep sequencing of these samples. And there's if we look on just a direct fraction of what we observe, here is the all species level taxa divided into the pan domains. On the left hand side, on the right hand side, it's the diversity pattern per sample, and the color states the individual. So there's at least 3 samples, mostly 4 samples per individual. So we see that there's quite a bit of variation in the diversity of the total pangenome species level distribution across all the samples. And then on the left-hand side, we can see from the sheer abundance of the reads is that bacteria is very much dominating, but there's a very strong fluctuation per filter regarding not only bacteria, but virus, fungi, and plant. And even some invertebrates were very nicely detected on some of the samples. So there's a huge variation in the total taxa what we find in the samples. Then when you look on the most abundant one, because there's a large fraction of the taxa which are very low in abundance, and if you go down in the species level, then there are a lot of caveats on how to include or exclude, and how to actually filter the data so that it is meaningful for the question. But as this is a pilot, we observe a huge variation, and we gain a lot of very diverse taxa. And on the domain level, the main observation is that the viral fraction is the one which, when you prune it, meaning that you exclude those viral taxa which in the classification are not that clear, then the viral fraction falls very low in most of the samples. But then there's a fluctuation of some samples with a larger fraction of viral content, particularly on phages, but also some of known DNA viruses. Nevertheless, on the heat map, what we can see on the right-hand side, very interesting parts. Here is the about 300 high abundant taxa in the pilot dataset on the y-axis. So you can see clearly on the heat map that there's a strong pattern of absence of several of the taxa, especially bacteria on some samples when you look on the compositional differences. So it is not only on what you detect from the metagenome, but then what fraction the composition actually tells varies a lot. So then going on different fractions of the data, for instance, looking at the plant exposure, we observed about 65 plant taxa species, and which we considered as a good candidates. And then especially 3 taxa showed very strong seasonal exposure differences. So on the right-hand side, you see a PCA showing the summer and winter months. So summer months are the red dots and with the red elliptic square, whereas the winter month samples are on the blue dots. And in the PCA, we can see that there's a strong seasonal differentiation among those good candidate taxa, which is very interesting on thinking how different is the exposure in the summer or in the winter months in Finnish environment, which can be very different. And some of these taxa, for instance, show traces of edible fruits or alga. Then when we look on the bacterial taxa, with the largest fraction of the biotic exposure, from there, we've got in this pilot data about 200 good candidates, high abundant taxa, and they give also a very strong seasonal differentiation. So on the winter months, a lot more diverse than in the summer months. And this again gives an indication that what we detect here is mostly indoor air exposures. And in that sense, the indication that in the winter time, people are more indoors or other factors, which we will address later on with the full cohort. But nevertheless, it's interesting to see already on the pilot data that there's strong seasonal fluctuation on these. Again, on different visualization, so that on summer months, the bacteria with a low abundance seem more diverse, and in winter there's a strong higher differences in a particular taxa in the data set. So again, and different ways to see the seasonal differences of the good candidate taxa. Then looking at the viruses, as mentioned before, in a total classification, there's a large selection of viruses, but when pruning the quality of the detection, it's clear that most of the viral traces are not that reliable. And with 32 good candidate taxa, we again see very strong seasonal fluctuation. And interestingly, on winter months, the diversity seems a lot stronger than in the summer months. Again, brings up the question on like, what is the quality of indoor air in winter or versus summer when people are more outdoors. So very interesting questions raising already from the pilot data. And also the interest on that there's a large fraction of phages found in the data set. And again, there's a seasonal fluctuation of the different taxa. Whereas when we looked at the fungi exposure, which is also very important abiotic fraction regarding our health, those good candidate taxa, we could not see any seasonal fluctuation. So interestingly, it seems very similar, the distribution of fungi exposure, at least in the pilot data, which is an interesting question, thinking what is our air quality and what are the different biological pandomains that circulate in our regular life. Here again on different ways to visualize these seasonal fluctuations, either looking on the effect size or in relative abundance. And also a bit of the invertebrate exposure. So even in 5 species taxa, we could identify in the data, although the taxa distribution is rather small, so I would not say too much on the seasonality. But when we look on the whole or the total biotic diversity distribution, as shown in the plot on the left, so we have the samples, which as a total show a gradient and not much clustering. However, we see that the pandomains do cluster differentially in the middle plot, showing on the bacteria, fungi, invertebrate, plant, and viruses, which we consider as good candidates, or at least reliable, based on the metagenome. And looking on the heat map of this again, we see an interesting presence-absence pattern across the samples. So these are the different ways to visualize and to understand how complex is the biotic diversity that we detect from these devices. And going another layer deep is in the looking on the exposure networks. Here as this the classified pandomain, and this is just to show that there's a very strong core network, which cannot see too much on the left, but then pulling it out from the network, there's a large fraction of the taxa that cluster much together, and then there's a different taxa which are fluctuating across the whole data set. So there's through network we can see that already in this pilot data most of the abundant taxa build a very strong core network. And to estimate on these exposures cofactors, the network helps then to visualize on here the color indicates the individual with the follow-up samples. So there's up to 4 samples per same individual, so same color. And then the shape shows that if it's on winter or summer months, when the year is divided into either winter or summer. And based on the high abundant, well-classified fraction of the metagenome, we can see that there's a lot of individual clusters where all the samples of the same individual, either winter or summer, or only winter, they cluster together. And then there are some bigger clusters, which also it's not included here on the plot, but because they are in 11 different city areas in Finland, they seem to associate into the same areas. So through building the networks with the well classified and characterized taxa, we can even find on these individualized networks of exposure. And so to summarize here, as this was a proof of concept and only the pilot data out of it, we can definitely conclude that using the UPass and including multi-site sampling is very feasible to do with pregnant women. Already as shown in the previous studies, but in this as well, we saw that the biotic exposure is very dynamic, either looking at the plant, bacteria, or invertebrate as well, and virus. Fungi is interesting, although the sample size in the pilot is small, so we need to verify that further. Nevertheless, this gives a very interesting view of our common environment where we are exposed to all these different pangenomian taxa. And obviously this allows later on with the data from the other site samples to do multi-site comparison, so that what are the likely origin and distribution of these exposures. And eventually then we can predict, very likely predict changes in individuals' health based on these traces of exposure. And a bit of the summary for the methodology, as the workshop series here is focused on the using and integration of omics, and especially working on the wearable device, low biomass metagenomes. The research question is very important that if you're looking on the presence, absence, or abundance of a taxa, of a pathogen, and that the method you're using, that they're aligned with your question. The classification is very challenging. All those who have done it even in the microbiome, we know that it's very challenging. And when you have deep sequence, low biomass sample, it gets even trickier because it includes environmental exposure, so a lot more diverse choices of taxa, where the reference database is very important, but often almost impossible to build to include everything. But you need to be aware on that, what are the knowns that you're actually looking at, and what are the knowns which you might not be able to detect, or if you can trust on that you get most of the biotic exposure analyzed, because it's a very complex and sparse data often, and it requires also specific statistics for that. So these are my short slides, and here I thank you for listening, and here are my colleagues and collaborators I want to thank within the consortium called Human Exposome Assessment Platform, HEAP, where we have been working on these same matters. Thank you.
Thank you very much, Ville. I think that this is a fascinating research topic. At least personally, I find it like that. It sounds like science fiction, but it is not. Like wearables are a reality, no, in our daily life, and it is really good to see research initiatives like yours taking advantage of it. So really good talk, and also really good the work that you have presented on the different methods for processing and curating this complex data, no? Highlighting the challenges that they are still there, no? Like the reproducibility that you mentioned, all these issues. So congratulations for this very nice project.
Thank you.
We have some questions from the audience. There is one specific question from Álvaro Torres asking about the potential privacy problems that can derive from collecting this type of information and analyzing this type of information. At the end, you are collecting genetic material, no? And he asked about which would be the best practice to respect the data privacy of this type of data. You are adopting special measures like the analysis.
Yeah.
Or, I don't know.
So I recommend to go to our consortium webpage, which is the in the European Human Exposome Network. You can find the HEAP project. But in practice, yes, the privacy is one very important aspect here, and as we know, in any medically oriented research is based on ethical permission, and like in this cohort work, the data is created and physically only allows to be in Finland, and this is the way that it is not shared publicly fully. Part of it can be shared, but not as the layer of being identifiable. And there's genetic data is one thing, microbe data is one thing, GPS coordinate is one thing. They're all very sensitive data. So you need to anonymize your data at some point when you're linking it together. But the main thing is that you have the computational infrastructure which allows you to work with sensitive data. I would say that that's the most important, because you need to have some place where you have capacity to combine and analyze the data. And it is a challenge with the large metagenome datasets. So it's, I would say cohort by cohort, you need to find solution. We have several cohorts in our consortia, and indeed, it goes cohort by cohort that what measures you need to take.
Okay. And another question from Mireia Bustos, is more specific about how to identify some taxa related to plants or bacteria for those species in which the genotype profile is not well known, or we don't have a reference genome. Do you think it's sufficient to analyze the genome of chloroplastids or mitochondria to identify them by barcoding, or long-range sequencing techniques more appropriate? It's a long question.
It's a good, long, and comprehensive question. Of course, we all would want to have a perfect data, but we also know that it doesn't exist. So you can address it several ways. The known unknowns are good, that when we know that there's, for instance, one domain of microbes that we don't have much of reference data, we know that we only capture a fraction of it. Then there are phylogenetic methods that you can build the pangenome, for instance, and then you can compare pangenomes. So that gives you an idea that if there's a difference in the variation of the pangenome, without actually knowing the different taxa within the domain, in more high resolution. That is one way. Then, of course, in the practical layer, it's can you use different type of sequencing and methods? So, for instance, short read, long read. Yes, if you have resources to do combined short and long read data, that may be a solution for you to build better resolution for your classification. But this is again depends a lot on the resources. And like if you think bacteria or virus, as a simple example, in bacteria you can always have a backup layer on 16S, but even though it doesn't give you so high resolution in the taxonomy, it gives you higher gain of the same sample, whereas in shotgun, for instance, you mostly get, in human samples, you get human DNA, and that can be a challenge then to create enough data that you can get higher resolution for the microbes. When you go out to other biological domains where we don't have so much reference data than human-centered studies, it is even trickier. So,
All right. And another question from Yuka Hintika is what is the required sequencing depth if you want to tap into the virome in the sample?
again, very good and very comprehensive question in the sense that one thing is what are the resources? Because as an example of this wearable device filter deep sequencing, in most of the samples, the viral fraction was very small when done as a shotgun. So if you have resources, you can go very deep, so create more data. But of course, that's not possible always. And then the question is if you could go on targeted sequencing, which helps you on this aspect, but it does not anymore allow you to see that the biome as comprehensive, because you only try to detect those you already know that your method will detect. So.
And another question is about the cost effectiveness of these methodologies. Are they feasible to implement in epidemiological studies with a large sample size? I guess that he asked for the cost of the devices and the analysis.
As we have seen is that even though devices may be costly, people are interested in their health, and like in this cohort, particularly all the pregnant women who were recruited, they were very interested in understanding what are their daily exposures. And in that sense, yeah, it's an individual matter on how much you would be paying if you could get this kind of a device as a citizen science project. Implementing as an epidemiological study, yeah, the devices are not cheap.
Yeah.
But as we all, for instance, use the pilot model, the prices are coming down constantly, and I would.
Around what per device? Around more or less?
At the moment, it's been like a bit below 2,000 euros per device.
Okay.
But you can reuse it. Everything depends on the battery quality and battery longevity. But I mean, I would be surprised that these devices would be in regular use in the near future, because the amount of data you get, and if you want to get an idea of exposures individually, the same devices have been used for let's say a region air quality estimations. So you tie the device into a pole on a street pole or somewhere, and then you let it run to collect information on the street, and building through a network of devices like an idea of a clean city or what is the exposure of the air. But yeah, it's very interesting. I wouldn't be surprised that these are put in the epi level in the near future, but at the moment,
Luckily.
yeah. At the moment, we're trying to prove that it's feasible on this kind of epidemiological, hopefully intervention studies, that we can actually find something that we could build a protocol for intervention.
Yeah. Okay, and I wonder, like, for most of the results that you have presented using these devices is about biodiversity or taxa profiles, no, across time by each individual. You mentioned that this is mainly related to infections as an outcome, no? It can be related, but I wonder which, like, did you explore connections between these biodiversity profiles and other external exposures like green spaces, or there are some studies studying biodiversity index in your residential area with your health? Have you looked into the connection with these possible external exposures and the internal or biodiversity profile that you have yourself?
Good that you pointed out. Yes, it's we are building a data set of the different type of environments in Finland and having more normalized data on what are the differences in the environmental exposures from non-human sampling. There's all kind of data available that's ongoing, and eventually, I mean, now the cohort work is in the halfway, and in hopefully in a year time, we can have the results to see that how it reflects to other environmental exposure information we have in different parts of Finland, and that, and including also the different body sites, so that we can start evaluating what might be a candidate on or what relevance they play in the exposure and regarding health.
Absolutely.
Um,
Yeah.
But
Okay, that's great.
it's a lot of work. So at the
Yeah.
moment, biggest bottlenecks are how fast to transfer data between computer clusters and you know, the common.
Yeah. Yeah. I'm sure that this is a challenge. Well, I think that's all, and it's now 5:30. So thank you very much. There are some comments congratulating for the nice talk. So thank you very much, Ville, and see you all in the next session next Wednesday.